Thank you for letting me talk about both of these studies. So now I would like to say we're going to get to why we have a Charlie Brown character. That's Pigpen. I'll get to that in a bit, but I do want to acknowledge my awesome partner for this work and co-author is Professor Dr. Claire Gwinnett. She's at Staffordshire University, and she is an award-winning forensic fiber scientist who's brought methods from forensics where knowing where the fiber came from is life or death in some cases to the ocean protection space. So let's cruise on here. Oh, there's Pigpen cruising around. So remember Pigpen. Okay, like I said, we're working on microfiber pollution with Rosalia Project. And when we, as part of the bigger picture, and when we heard about the problem, we set out and made 3 goals as it related to, in particular, microfiber, but in a lot of ways, we're doing microplastic along the way. One was to learn and discover more about the problem. We learned about it in 2013, which was very early. There wasn't a lot of information about it. The fact we're having whole conferences now is amazing. We wanted to develop a solution. And again, full disclosure, we're the makers of the Coraball, the washing machine microfiber catcher. And we wanted to see if we could inspire innovation and development in all the myriad industries and subjects in which we don't have experience, and we're doing that by spreading the word. So back to that Hudson River work. We did it in 2016, which gave us the results that I talked about in the last talk, unexpected, and indicated additional sources. So we went back in 2019, and this time we studied more of the river, more environments. We took air samples at 2 heights, my inhalation height for the whole river. And while we were on American Promise, our research vessel, which was Ambrose Light to Albany, so the mouth of the river to the middle of the river effectively, we had a second one up at 9 meters. There's the 1.5 meter air pump. We took soil samples where along the river where there was some kind of sand or soil or some kind of terrestrial situation and not either cliffs or Manhattan City. And here's Coney Island. We retook surface samples. So there's Claire throwing out the bucket. And we also used a Niskin bottle to take mid-water column samples and just above the seafloor, as well as took some samples for nanoplastic work. So this is the wastewater treatment plant study I mentioned. I'm not going to share results about this yet. We haven't sort of finished all this yet. But the way we did our wastewater treatment plant study was we tested at the 0 marked on the chart outflow, so that's 0 here, of 3 different plants along the river, a rural, a suburban, and then very urban right there between Manhattan and Brooklyn. And then we did the whole water column, so surface, mid, and just above the river floor. And then always with the same current, so always in a max ebb, we started up current and we went from 2 miles, 1.5, 1.5 to 0, and then same thing, half-mile increments out. That's going to be important when I talk about this, which is what this talks about, is something that we were thinking about the big picture. So we did all those work to figure out, like, are there relationships between microplastic in the air, the soil, the water, the surface of the water with the middle of the water, and that kind of thing. But we also were looking at this sort of big picture of microplastic science, especially through the lens of, could it be done by citizen scientists? There's so much we have to learn. And we wanted to know primarily, are we contaminating our own samples? Considering that what we're getting from the river, the air, and the soil is made from clothing, and we are wearing clothing while taking these samples and processing them, what is going on? And so I'm going to get right to the results of that study. This is published in Marine Pollution Bulletin last year. There's the citation, you can email me. Okay, so this is first and foremost, what we wanted to know is when we used all of the protocols, all of the anti-contamination protocols, how much good was it doing? The way we checked our work effectively was we took samples of every garment, every bag, every piece of the boat, sail bags, lines, everything. So we had a catalog of all of the fibers, this is in particular fibers, that could contribute to contamination. And when we found a fiber in one of our environmental samples, it was first matched against that catalog. If there was a match, it was considered from that thing, a t-shirt, whatever, and considered contamination. If it wasn't, and it also wasn't in an ambient air blank, so if it was an air blank, that was considered ambient contamination. So if it wasn't in either of those, then it was considered a pollutant. Now, this, I have to say, surprised me. When we used all of the state-of-the-art anti-contamination protocols, 33.7% of what was in our sample was traceable to specific known sources of local, like our clothes or on the boat, contamination. So that was more than I expected, I have to say. But then we did a specific study where we didn't follow the rules on purpose. And we split it up between the field collection side, which for us was on deck, and the processing or the vacuum filtration side, which was down in a normally cleaned galley with the doors closed and windows closed and everything. And so we first isolated the deck, where basically we didn't follow any of the protocols. We did all the things wrong. We stood upwind, we dropped lines into the bucket and didn't throw it out, stuff like that. But we did it right in the galley. And then we reversed it, where we followed all the rules on deck and we didn't follow rules on the galley. We let people hang out and talk. We turned on the fans, that kind of thing. And it was a lot of contamination, around 70% on either side, depending on it was deck and galley. And what was also interesting was it was essentially the same amount. That there's a lot of talk in studies about protocols in the lab, but there's not often, or there hasn't been historically, talk about protocols collecting samples. And the reality that we found is that you could have 70% of what's in the environmental sample could be contamination rather than pollution without protocols. So just sort of isolating those. There is a big difference between using anti-contamination protocols and not, whether you're doing it, you're considering the actual collection itself at the shoreline or from a boat or wherever it's happening, or from a lab. And we are obviously testing a portable lab. And so my conclusion here is that we're all Pigpen, but instead of walking around in a little cloud of dirt, we're actually walking around in a little cloud of microfiber. Because then we're like, okay, what clothes contributed? And here's all the clothes that contributed. It's basically all the clothes that we were wearing. And then we thought, okay, whose clothes contributed? And we did have uniform tops to sort of control this and for the visual. But the reality is that even our captain, who was always behind the wheel for collection, we were always facing into the wind. So he was downwind of all the samples all the time. He wasn't even allowed down below. His surf shorts made it into the samples. So it didn't matter where people were on the bow, doing sampling, sort of amidships, doing Niskin bottle, whether they were down below, or even they were the boat ops team in the aft, in the aft part of the boat, cockpit, not even mixing with samples. It came into, we had contamination from all of that. Now, remember, I'm also thinking about all this, not just to make our science more robust, but also, can we do this with citizen scientists? And I think what we learned is that, in fact, we can, but we are going to need to take some action. And that action includes addressing this contamination factor. And we were wearing 100% cotton t-shirts, but it does turn out that cotton is a big shedder, like a lot. And I think that it would simplify the science to wear a very, very low shed garment that covers the majority of a person, and that is a color that we don't normally find. And I did sit in on one of the presentations before me. It looked like there was red and blue, and we have lots of evidence that the vast majority of the fibers, at least that we're finding in the Hudson River, are red, blue, black. Well, blue is number one, blue, black, red in that order. And this kind of colorless orange, we barely find orange fibers in our samples. So I'm guessing that that's the look we're going for. One last thing I want to share is just something that we did in this paper as well. And it's using a polarizing light microscope to help us make the identification. Because if you think about it, it's great. It's okay if we're having contamination if we can find it. So I'm not worried about science and robustness and that if we know that it's happening, but it takes a lot of time to say, oh, a blue fiber, is this a blue fiber that somebody was wearing? So we also were looking at efficiency and also trying to not have to rely on FTIR and Raman spectroscopy quite as heavily. And so Claire and her team brought this using a polarizing light microscope across to the field. And we found that we could make an identification of the type of fiber to the material level, like polyester, acrylic, nylon, or whatever, in over 90% of the items of interest, let's call it, which is pretty incredible and way more efficient because we also timed how long that took compared to how long it takes to use an FTIR. And it was 3 minutes compared to 8+. And a polarizing light microscope, we just got one for $2,000. So there's some interesting, actually, I found one for $2,000 since this slide was made. So I'm going to leave with just one last reminder of how vulnerable our clothes are to breaking. Here's a whole array of magnified clothing from acrylic to two polyesters on the top, polyester cotton blend that's high shedding. We've got, interestingly, the nylon outside of a wetsuit, a pair of yoga pants, nylon spandex, linen and rayon, and 100% cotton. And just a reminder that this stuff is small. This is a real hard problem to fix on the back end as far as cleanup. So I think it's one that needs preventing. And I like to share some solutions. I don't know that we quite have time to go through them. So here's our thoughts on solutions for washing emissions. Our ideas on dryer emissions, where I think we need a lot more research on this, especially as it relates to air drying. I'd love to know. And finally, for wearing clothing, which I really think is all about making our clothes more resilient in the first place and understanding what that takes. So my autocorrect changed Coraball to cornball, which is horrific. And I will stop sharing. And if there's time for questions, might be close.
We do have time for questions.
Okay. Related to the Coraball, we were curious about how big is the current market for the Coraball, and can you share any numbers on sales?
Yeah, we have sold now, we're just rounding on about 90,000 of them in over 62 countries worldwide. And that brings us a lot of hope that people are willing to take some action to reduce this pollution. The Coraball, since it was introduced, also has been shown to reduce the shedding that happens by your clothes in addition to collecting fibers. And my take on it after a lot of years, 5 years since the Coraball launched, is that when people learn about the problem of microfiber pollution, it resonates and they are willing to be part of solutions, whatever that is, using cold water, washing with 3/4 to full loads, using a Coraball, using an aftermarket filter. And while the regulatory side and the innovation side in laundry machines spins up, because that's obviously taking a long time, the fact that we can do a little bit with products like the Coraball or techniques like cold water, full loads, that kind of thing resonates with people.
I would agree. I think people want solutions.
Mm-hmm.
Right?
Me too.
We have time for another question.
Okay. One of our attendees was curious about the long-term effects of inhalation of these fibers and what are the contaminants that people are inhaling. And finally, what clothing has more than others, like maybe sheds more than others in creating the pig pen cloud effect?
I can't speak on the human health side. It's not my area of expertise. There's a paper that I did address the inhalation part during an air study in London. Its lead author is Stephanie Wright with a W. And so I refer you to look at some of that work. It's an important question. The answer to that is an important one. So I certainly appreciate it. Then on the clothes. So again, it all started with washing machines. And then there's like a little moment of dryers like 2 years ago, and that's growing. And now there seems to be also a growing recognition that the wearing of clothes is a contributor. I mean, it really was a lot of years where all anyone talked about was washers. So the drying and the wearing component is the newest part of this science, and it's very, very new science. I don't have a great answer, but I have a magnitude, is that we were getting contamination in samples that were exposed to the air for conservatively less than a minute, but some of them were open to the air for less than 20 seconds, and they were getting contamination in them. So to me, there's a lot coming off our clothes, and it's hard to measure for the air. There's some work out of the Plymouth University lab. I think there's a DeFalco and Imogen Napper. Those two have done some really interesting papers moving towards understanding emissions while wearing. Right now, though, the majority of the information on emissions is, as it relates to specific fabrics, is in washing machines. So here's another area of opportunity for research.